Okay, in Work Package 1, we are trying to find drivers for Xylella fastidiosa epidemics in Europe under current conditions, but also in future scenarios associated to climate change. So we have focused in different activities. We are trying to find new variants of the bacterium that could be tested using different approaches. We have sentinel plots that are plots located in different areas in Europe, South, North America, Central America, and South America to test the presence of the bacterium in this area and looking for new variants. And we have identified in several of these sentinel plots some new variants of the bacterium that was not observed before. In parallel, with the same objective, we have the collaboration with the botanic gardens in Central and South America, similar approach, trying to detect bacterial population that were on those countries, and we were also able to identify variants in the bacteria in areas where the bacterium was not identified before. So another approach was to find the host range. So which host can be infected by the different strains of the bacterium, different species, on different genetic variants in control conditions. In this case, we work in biosecurity structure, greenhouses or grow chambers, because we are dealing with a quarantine bacterium and we have to work under this condition. And we have identified a very complex situation because when we want to estimate the risk for a certain host, we need to consider the variant, the subspecies, or even the sequence type of the bacterium to really assess the risk for this particular host. On the other side, when we want to test the risk of a certain subspecies or a certain strain, we have to look for the specific host range, which plants can be affected by this strain. This work involves not only cultivated plants, but also like olive, almonds, or grapes, but also some other hosts that can be important for certain regions, like carob or persimmon in the Valencian community that are planning to be used to replace those affected almonds in this region. Then we have also a set of experiments in which we evaluate how genomics analysis can help, for instance, to identify the origin. So when a strain was identified in a certain area, and for instance, using this approach, we could identify the origin of some of the outbreaks in Europe, the origin of the strain that was introduced in a certain place, like in Portugal. So then we also have developed the models to estimate how soil properties or climatic conditions are more favorable for the establishment of the bacterium. So if Xylella fastidiosa is introduced in a certain area in Europe, we know the risk for this strain to be established. And finally, we also assess how different climatic conditions could affect the future development of the disease. In particular, in grapes, we have analyzed the combined effect of temperature, CO2 concentration, or water regime in the development of the disease, trying to simulate future scenarios for the disease that are associated to climate change. And in summary, this is what we have been doing in Work Package 1 in the BeXyl project.